This video is about mitosis and a little about how mitosis relates to cancer. So let's meet Judy, who loves to sunbathe. And in this case, we might ask the question, well, how is sunbathing related to skin cancer? And why is skin cancer even a major problem? So to tackle the first question, sunbathing is related to cancer because ultraviolet radiation from the sun is a carcinogen, a cancer-causing agent. What that does is often mutates DNA or changes that DNA to cause cells to copy themselves uncontrollably. So the cells divide and begin that process of cell division in an uncontrolled sort of way. And so those skin cells start to multiply and grow. It's not a major problem until it starts to spread to other body organs, and then it becomes a major problem. One of the ways your skin works to defend yourself against ultraviolet radiation is just the programmed cell death. That rather than reproducing mutated cells, those cells just die, and as a result, the skin peels. And so we often see that when we're sunbathing. Here are some things to look for with moles and to see if they're cancerous. Now, benign just means that they're not going to spread and grow. Malignant means that they will spread and grow. So it's the malignant ones that we're really super concerned about. So these are cancer cells that could cause major problems. One thing to look for is asymmetry. So we're just going to follow the ABCDs. So asymmetry means that it's not the same on both sides. Border means that the border is irregular, it's uneven, jagged, or rippled. The color means there's more than one type of color, like there's two tones to it. And the diameter, if it's over 1/4 of an inch, it's too big and it could cause cancer. What's happening underneath is that those cells are dividing by mitosis. That's the process of cell division for body cells. And that cancer is growing. So from like picture A to picture E, the cancer is getting larger. But at picture E, it's significant because now that it gets to the bloodstream, those cancer cells could travel around the bloodstream and begin to infect other organs. So what goes wrong with skin cancer? Well, to understand that, we need to understand the process of mitosis and the cell cycle. So this is what the cell cycle looks like. It's the life cycle of a cell, that it spends most of its time in interphase, in fact, 90% of its life in interphase. In fact, some body cells never get out of interphase, like brain cells or muscle cells. Those cells never divide. So during your whole lifetime, they just spend time in interphase, where they just do interphase things, where they grow, the DNA gets replicated, and they do normal cell processes. So some cells spend all their life in interphase. Then we go to the next phase, mitosis, which is just cell division. So part one of interphase is called the G1 phase, and that just means growth. So the cell is just spending time growing and copying those organelles and getting larger. S stands for synthesis. In this phase, the cell is synthesizing DNA. So here's where the DNA gets replicated and copied. And then the G2 phase is when the cell is growing some more. After the G2 phase, then mitosis begins. During the cell cycle, there are some checkpoints to see, will the cell continue in this cycle or will it stop? The stoplights represent those checkpoints. The first stoplight is going to evaluate the growth. Is the cell large enough to divide? If not, it stays in the G1 phase. If so, then it'll continue on through the rest of the phases in the cell cycle. The second stoplight after the G2 phase mostly is checking the DNA. Was the DNA replicated correctly? Are there any mutations in that DNA? If so, the cell stops. Maybe it dies. If not, the cell continues through the cell cycle. During the cell cycle, in anaphase, there's another checkpoint, which is checking to see, are the chromosomes being divided equally? In other words, will the two new cells have the right number of chromosomes? If so, the cell continues on, and if not, everything comes crashing down. Now, these checkpoints don't always work. And it's when they don't work that we are in the most danger of getting cancer. To understand mitosis, mostly it's going to be the division of the chromosomes. Chromosomes are just DNA. Now, normally your DNA in your nucleus is just a big blob. It's hard to tell what's what. So right before the cells divide, the DNA condenses into chromosomes. That keeps it organized. And it's organized because it wraps around these things called histone proteins. So here's our chromosome. The chromosome has been duplicated, replicated. That's why I have two chromatid. The center that's holding the two together is called the centromere. Now, sometimes the problem is we count our chromosomes by the number of centromeres. So, for example, humans have 46 chromosomes. But when those chromosomes are replicated, we would say you still have 46 chromosomes. Why? Because we're counting centromeres. But you actually have 92 chromatid. Let me show you what I mean. Here's a chromosome, and in the middle you can see the centromere. But now here is a replicated chromosome. So I have sister chromatid, but it's still just the one chromosome because I'm counting that green dot, the centromere. Here are 6 chromosomes across the top. There's 6 chromosomes across the bottom. The difference is the chromosomes across the top have the sister chromatid, those replicated parts to them. So let's look at mitosis, dividing the cell's DNA. And then the second process is cytokinesis, where we divide everything else. So first we're in interphase, just growth, DNA replication, normal cell processes. If we pass those checkpoints, we get to mitosis, where the cell begins to divide. So prophase is part 1, where the chromosomes become visible. The nucleus disappears. I remember these stages by the acronym IPMAT, IPMAT, I-P-M-A-T. So I stands for interphase, P stands for prophase. So prophase, the chromosomes become visible, the nucleus disappears. Metaphase, the chromosomes line up in the middle. Anaphase, they begin to move away. So I remember metaphase, middle, anaphase, away. The chromosomes move to opposite sides or poles. Telophase, I remember 2. So metaphase, middle, anaphase, away, telophase 2. In this case, 2 new nuclei form. Nuclei is just the plural for nucleus. I think of this kind of like gym class. In interphase, everyone's just kind of mingling around. But then the teachers come out, and that's like prophase. They blow the whistle, and the classes begin to sort and organize. And in that case, you can begin to see the classes are distinct. Like in prophase, the chromosomes become distinct. Then the teacher says it's time to play a game. Let's say kickball. To play kickball, you have 2 team captains. Those are like the centrioles. And then you'll have everyone else line up in the middle. And the captains begin to pick, and they're trying to pick evenly. So just like the cells dividing the chromosomes evenly, the captains are dividing the teams evenly. And after the captains pick, like in anaphase, then we have 2 new teams, like we would in telophase. To get those chromosomes to divide evenly, the chromosomes will move on what's called spindle fibers, and they move toward the centrioles. That's kind of like the team captains in my example. Now, plant cells do not have centrioles, but animal cells do. So here's a cell, and you can see it's in metaphase, and the chromosomes are lined up in the middle, and we should get a blue, a yellow, and an orange moving to each side. But what if that's not the case? What if that checkpoint isn't working? What if the 2 cells divide and we get an uneven number of chromosomes? In that case, the one dies because it doesn't have enough chromosomes to live. The other has an extra one. Now, if that yellow chromosome has genes on it that slow cell growth, that cell also dies. But if that yellow one has genes that increase growth, that is how cells can become cancerous. So sometimes cells become cancerous because the DNA, and there's a problem. Sometimes the cells become cancerous because the checkpoints aren't working. And if one of those checkpoints isn't working, the one that manages how many chromosomes are in each new cell, then we could have the chance of cancer like this cell is showing. I hope that helps a little bit with mitosis and how it relates to cancer.